Hello dear friends, welcome to this video on our YouTube channel. In this video we are going to be looking at the correction of the Northwest the 2025 guys Northwest Advanced Level Biology mock paper 2. We shall be looking at a question that was based on genetics. Okay, now guys, if you're new to this channel, don't forget to subscribe to the channel, because in the channel, friends, we have a lot of contents for you. So long as you are a science student and you are an O-level student, we have enough contents for you. You can only have access to all those contents only when you have subscribed to this channel, friends, and share the video out with others so that it's going to motivate the YouTube algorithm now to recommend our videos to so many audiences. Thank you very much, friends. Let's go on. The first part is saying that in a tabular form, differentiate between the following pair of genetic terms. We have codominance and incomplete dominance. The next, you have gene mutation and chromosome mutation. Okay. Then the next part, friends, talks about genetic crosses. Let's look at the first, guys. The question has specified that we should do it in the tabular form. So let's insert a table. That's it. We have this way we have codominance, and here we have incomplete dominance. So friends, the best way in which I recommend you guys to do this is by the definition. Okay. Now guys, how do you define codominance? It is simply a situation where for two or more alleles, friends, express themselves to the same extent, mostly in the phenotype, in a phenotypic extent. Okay, and in such a case, guys, intermediate species are being produced. It's just like a cross between a green flower and a pink flower. The offsprings in the F1 have blue flowers. You can see that the offsprings produced have different phenotypes compared to the two original parents, right? So that's a case in which the two genes now are trying to express themselves to the same extent. In such a case now, an intermediate phenotype is being produced which is distinct from both parents. So in that case, guys, that's a case where you can see codominance occurring. Okay, let's look at incomplete dominance, guys. Again, by the definition, incomplete dominance, friends, is simply a situation wherefore the effect of the recessive allele is not completely masked by the dominant allele. It's just for instance that let's say that if we have a case, if we have two alleles, right? If we have two alleles, under normal circumstances, friends, we either expect that all the offsprings produced in the F1 generation after a cross between the two guys have to give us what, either one of them that is amongst either the parents, or we have an intermediate species compared to what, just like in the case of codominance. But friends, whenever we have a case in which we see both the characteristics portraying themselves, guys, in such a case, that is known as what, an incomplete dominance situation. Friends, note that incomplete dominance is midway between codominance and complete dominance. Yes, so don't ever mistake codominance for incomplete dominance, guys. This incomplete dominance, like I said, is midway between codominance and dominance and a complete dominance situation. Okay, so guys, that's it. The next part was also to differentiate between gene mutation and chromosome mutation. Okay, again, friends, by the definitions, let's look at gene mutation, guys. So friends, gene mutation simply refers to a change in the structure of the DNA in a single locus. Keep in mind, friends, that the exact position wherefore a DNA molecule is found is called what? Its locus. Okay, great. It is called its locus. That's the exact location, friends. That's the exact location where it is found is called its locus. Okay, let's look at chromosome mutation, guys. So friends, chromosome mutation simply refers to a change in the amount or the arrangement of chromosomes. Okay, so guys, keep in mind that in a normal individual, there are what, guys? There are 46 chromosomes, which we have 23 pairs. If there are more, it is an issue. If there are less, it becomes what, guys? An issue. So in that case, guys, it is mutation, specifically chromosome mutation, where for the number or the structure of the chromosomes, friends, have changed from the number 46 that we all are supposed to have. Okay, so guys, that's it for this section. If there's something you don't understand, or there's something that my explanations are not able to make you understand, friends, let me know in the comment section down below, friends, so that I can explain more for you as soon as possible. Okay, Let's look at the next part, guys. Okay, guys, the next section, the next part is that a farmer crossed a cocoa plant that has yellow long pods with another having red short pods. All the F1 plants produced had yellow long pods. When he crossed the F1 cocoa plants with cocoa plants having red short pods, the following were obtained. We have 483 cocoa plants with yellow long pods, 103 cocoa plants with yellow and short pods. We have 472 cocoa plants with red short pods, then 93 plants with red and long pods. Okay. Then the Roman one is saying that we should use genetic crosses to explain these results as fully as possible. Then the next part wants us to, using the figures given above, should calculate the crossover value between the genes for the nature of pods and the color. Okay. Now, guys, let's start with the very first part. The first part of the equation, friends, is simply saying that if a farmer cross a cocoa plant that has yellow long pods with another having red short pods, all the F1 plants produced had yellow long pods. So, guys, since all the F1 plants produced had yellow long pods, it therefore means that the gene for yellow long pods is dominant over the gene for red short pods. As such, guys, we can simply go ahead now to make our statement. But before then, guys, we have to be able to tell if there is linkage in this gene and if there is crossing over or not. Because once you fail to be able to detect if those two features are present, guys, you have high chances of failing this question all. Okay. Now, guys, let's look at the figures that they've given us here, guys. Let's examine the figures. So we have the figures 800 and 483. We have 472, 103, and 83. So, guys, if you divide these figures here by the smallest, namely 93, if you take every number here and divide by 93, guys, and you run your answer to the nearest whole number, you're going to see that the ratio that we are going to have, friends, is going to be 5:1:1, right? And we can see that, what, guys, that this is not a Mendelian ratio of 9:3:3:1. It ever means that there is linkage. Okay, whenever you take the ratio of these numbers, guys, and it gives you 9:3:3:1, therefore means that this is the Mendelian ratio. It ever means that there is no linkage. Okay, there is no linkage. But since this our ratio has not given us 9:3:3:1, namely it has given us 5:5:1:1, it therefore means that this ratio is not the same like this, means there is linkage. Okay, now, guys, let's see if there is crossing over. Okay, now, guys, let's look at the parents. Do you see that the parents in this case, we have yellow long pods, right? Crossing with what? Red short pods. So whenever the parents are occurring in a very, very high number and the offsprings are occurring in a very small number, means there is crossing over. So in that case, guys, do we see that we have 483 cocoa plants with yellow long pods, right? It is a parent. Then we have red short pod is also a parent, right? The parents are occurring in a very high number compared to the offsprings, means in this case there is linkage. Okay, there is linkage. So in this case now, friends, we have both linkage and crossing over. That's good. So now, as I was saying, guys, by the explanation, the parents are occurring in very high number means there is crossing over. Great. So, for instance, it's very, very important that you should know that there is crossing over. So in that case, guys, since the equation told us that the offsprings produced had yellow long pod, it therefore means that the gene for yellow long pod, like I said, is dominant over that for red short pods. As such, guys, we can let capital Y to represent the allele for yellow. Then we are going to let our small y represent the allele for red, since red is recessive while yellow is dominant. So we are going to let capital Y to represent the allele for the dominant gene. That's the dominant allele, okay, the dominant, which is yellow in this case, since all the offsprings in the F1 had yellow long pods. Similarly, guys, we're going to let capital L to represent the allele for long pods and small l to represent the allele for short pods. Great. So, guys, just by these statements, guys, if there are enough marks, you've scored 4 marks already. Okay. Then now, guys, as such, guys, we have to go now straight to the parental phenotype. So the parental phenotype, guys, are what we are crossing what a yellow long pot and a red short pot. So we are going to have long pots, yellow long pots times red short pots. Great. Then now, guys, we have to go now straight to the parental genotype. Since we have said that there is crossing over, right? There is since we have said there is linkage also, so the genes have to be linked. Okay. Now, guys, what is the genotype for yellow long pots? What is that for yellow, guys? It is what? It is Y, right? Wait, it is Y, capital Y. But since we are talking about genes, right? Genes must always occur in pairs, okay? In pairs. We are going to have, for yellow is capital Y, capital Y. For long is what? Capital L, capital L. For red is what, guys? Is small y, small y. For short is what, guys? Small l, small l. Great. So we are going to have y, l, l. That's capital Y, capital L. Crossing it with small y, small l, small l. Okay. So now, guys, notice that this is homozygous for yellow long pots. This is what? Homozygous for red short pots. What will happen if we rather had, but if what we have is capital Y small y, capital Y small y and capital L small l, it will still be what? It will still be yellow long pot, but in this case now a heterozygous state. Okay, this is the homozygous state because all the alleles in this case are identical. That's all the pair of genes are identical. Great. Okay. Then now, guys, since we earlier said that there is linkage, it ever means that we have to put each of these our genes now, our genes now in what? In linked traits. So in that case, guys, we are going to take capital Y, capital L and put it on a locus. So we are going to have it like this. So we are going to insert capital Y, then we are going to have capital L down here. Okay. The next again now, guys, we are now going to have, if we do the same for all of them, we are going to have and capital Y again, then we have capital L. Then we do the same this way, guys. We do the same this, guys. We are going to take small y, small l, small y, small l. So, guys, this is how we represent genes that are linked. In this case, guys, there is linkage. Okay. So, guys, at this point now, all we need to do now is to do the crosses. But you discover that we have pairs of genes on this way and pairs of genes this way. That's identical genes for that matter. So in this case, since we have capital Y, capital L, capital Y, capital L, small y, small l, small y, small l, we are going to come to this first parent, right? We simply take one of the genes. We take just one, come to this one, take also one and cross. Okay, guys, let's go to the next slide and see what happens. Let's go. Okay, friends, I've simply copied out the things that we are going to need in this cross. Okay, let's insert all we need. Okay, we need to go straight to the gametes. How do you form the gamete, guys? The gametes have been formed by putting each of the genes we have to cross in a circle. So we are going to have them in this case. Like I said previously, guys, we are going to go to each of the various sections. We take just one of them since they are identical. We come to this. We've taken this, right? To this order, we have taken this as well. So if we cross now, we see that we are going to cross these two guys, right? And when we cross them, we are going to have what? We are just going to have just these guys. We take this is it, and we have this is it. So, guys, how do we call this? We call this what, guys? The F1 genotypes, okay? This is the F1 genotype. How then do we define the F1 phenotype, guys? Do you see that in this case, since capital Y represents what? The allele for what? Yellow, right? Capital L represents what? The allele for long. So in this case, all of the offsprings are what? Have yellow long pots. Don't be deceived by this small y and small l, because small y and small l, guys, are recessive alleles. They are recessive. So capital Y, capital L are dominant. So in that case, guys, it is going to mask the effect of this guy. So all the offsprings are going to have what? Yellow long pots. So this is the F1 phenotype. Okay. So now, guys, we have simply proven what the first part was saying. Let's now see how. Let's now look at the next part. The next part is saying that all the F1 plants produce hard yellow long pots. Good. Then they're saying that when he crossed the F1 cocoa plants with cocoa plants having red short pods, the following were obtained. Okay, so we need to cross this F1 plus now with cocoa plants that have red short pods. Okay, let's go now, friends, to the next slide and see what happens. Let's go. Okay, friends, let's go ahead. Okay, so as I read earlier, guys, we need to cross the F1 plants with plants having what? Having red short pods. Okay, so guys, we will simply go straight to the parental phenotypes. The parental phenotype, guys, we are going to be crossing what? Yellow long pods, right? Which was the F1 we had with what? Red short pods, as the question is saying. This is when he crossed F1 cocoa plants. What are their phenotype, guys? All of them had what? Yellow long pods, right? When he crossed F1 cocoa plants with the F1 cocoa plants with cocoa plants having red short pods. Great, so this is it, guys. Then now, guys, the parental genotype, guys, as me going to be this. Okay, so yellow long pods. This, what is this, guys? This is simply the result we got after this are the F1 genotypes, right? Great. So the F1 genotype, as the question said, they said when he crossed the F1 cocoa plant, these are the F1 genotypes. Then we have to cross it with what, guys? With cocoa plants having red short pods. So this is going to be the gene for red short pods. Okay. Then now, guys, at this point now, this is where crossing over occurs. Okay, this is when crossing over occurs, and crossing over occurs, guys, between this unsimilar gene and this one. Okay, this is where we are going to see crossing over happening. So during crossing over, guys, there's going to be an overlap of these our genes. Okay, they're going to overlap and exchange segments such that the resultant alleles produced are going to be genetically distinct from each other and from their parents. So, guys, crossing over, guys, this is crossing over. Okay, let's now go on now. During crossing over, guys, now we are going to see this one remains the same. This our gene remains the same. This other, guys, also remains the same. Okay, this one also remains the same. Then now, guys, these two now are going to have the crossing over happening. So this one now is going to bend in this way. This one also bends such that this is what is going to be happening. Okay, this is the crossing over happening. So now after crossing over now, guys, these two segments now are going to split at this point. Okay, they are going to split and we are going to have this gene coming out and this gene coming out. Okay, so now, guys, I hope you understood what's meant by crossing over, right? This is the process of crossing over that actually occurs. So we split it like this. We split transversely from top to bottom. Okay, let's go to the next slide, guys, and see what happens. Okay, friends, after the crossing over, guys, we have to just form our gametes. By forming gametes, guys, we simply take each of the genes that we have and put them in a circle. So after crossing over, guys, you notice that this is the gene that we have, right? This and this, they have exchanged what their segments. They have simply exchanged their content. So now we have capital Y and we have capital L here. Great. So in this case now, guys, let's do the crosses. When we cross this guy and that guy, what are we going to have? We are going to have this gene, capital small y, small l, and what? Small y, capital L. Good. If we cross this other and these guys, what are we going to have? We are going to have this gene, small y, small l, and what, guys? We have small y, small l. We also cross this and this other. What are we going to have? We are going to have capital Y, small y, capital L. We also cross these two guys, friends. What are we going to have? We are going to have capital Y, small y, small l. So this, how do we call these guys? We now call this the F2 genotypes. Okay, these are the F2 genotypes. Okay, now, guys, let's go to the next slide now to see what happens. So these are our crosses. These are the crosses. You simply have to interpret the results. Okay, let's go to the next slide. So, guys, these are the results that we get after crossing, right? So these are the F2 genotypes. Okay, let's now get the F2 phenotypes. How do we get the F2 phenotype, guys? We simply interpret our results. Do you see that we have small y, small y, small l, capital L, right? Capital L surpasses what? Small l. So what allele was small y representing? Short red, right? Red, because it is representing color, so it is red. So in this case, what is the phenotype of this guy? It is what? It is red long pods, right? Red because we have small y, long because we have a capital L. At least we have capital L, we have small l. So capital L is dominant over small l. So we have 93 red long pods. We have to use the figures that the question has given us. Okay, the figures. So we have 93 red and long pods. So this is it. Then the next, guys, you see that we have small y, small l, small l. So this is what? A recessive gene. So it means this is going to be red and short, right? Red short pods. And how many of them are there, guys? How many of them are there? We have what? We have 472. So we're going to have 472 red short pods. In this way, guys, we have capital Y, right? So it's already yellow. Then we have capital L, so it's already yellow and long. So this has what? Yellow and long. But how many of them are there, guys? How many? We have 483. So we're going to have 483 yellow long pods. Then in this case, guys, we have capital Y at least, right? So it's already yellow. Then we have small l. Everything is small l, so that's what? Red. So we are going to have what? It has to be red. So we are going to have, that's a short. Sorry, short. So we're going to have 103 yellow short pods. So this is what, guys? This is the phenotypic ratio. This is the F2 phenotype. The question did not ask us to give the phenotypic ratio, so we can leave that aside. If you give it, good, that's extra bonus for yourself. But I won't give it, guys. I won't give it. If you have been asked to get a phenotypic ratio, all you need to do is simply to divide these figures you have. In fact, earlier on, guys, we saw it, right, that it was 5:5:1:1, right? That's the ratio that we must get. So this is it, guys. If they ask to give their various probability, guys, what you have to do is to divide each of these values by the sum of the two of them. If you sum the two of them, so well, guys, I'm sure if you sum the two of them, right, and divide by each of them, it's going to give you the probabilities. Okay, yes. So you simply divide 93 over the sum of all of these figures. But don't punch your calculator, guys. Don't punch. Keep it as a fraction. Okay, guys, this is it. We are done with that part, with the Roman 1 of that part, and it was for 12 marks. It was for 12 marks. So all the 12 marks have been scored so far. The next part is saying that we should use the figure given above to calculate the crossover value between the genes for nature of pods and the color. Okay. Now, guys, let's note that the crossover value, guys, is to be defined as the number of recombinants over the total number over the total population present multiplied by 100. Okay. So now, guys, let's go ahead. So the number of recombinants, guys, are simply what the offsprings produce. Do we see that in this case we are crossing what? We are crossing, in this case, guys, you see that the parents are what? Yellow long pods, right? And the parents are yellow long pods and what? Red short pods. What are the offsprings, guys? Anything that is not yellow long pods and not red short pod is an offspring. So we have red long pod, this is an offspring. Then we have what, guys? We have yellow short pods, so this is also an offspring. So, guys, the crossover value or the COV value has to be, we are going to have 103, that is the value in this case, plus 93 over the sum of all of these figures times 100. If you punch this so well, guys, we have an answer to the nearest whole number of 17 units. So, guys, this is the answer for this question. Now, friends, one thing they might be asking you again is to list the significance of this crossover value. So, guys, I will give it to you back, guys, as an assignment. Just simply put in the comment section down below your answer. What is the significance of this crossover value? If you don't know the answer, guys, simply let me know in the comment section that you don't know, so that I can tell you, so that when you get to the exam hall, guys, you will not be left out. Okay, guys, thank you so much for watching the video. Make sure you subscribe to this channel if you want to have more great content from us, guys. Visit the channel description, guys. You're going to find links to our WhatsApp group, and you are also going to find the link to watch other videos. Have a blessed day, friends.